Hello, and welcome to today's webinar titled Future Trends to Control Risks from Extractables and Leachables, hosted by Biopharma Asia Magazine and presented by Jason Creasy, Managing Director at Maven Extractables and Leachables Limited. Hi there, Jason. Thanks for joining us today.
Hi, nice to see you all today.
Yep, it's a pleasure to have you, so thank you for joining us. And today's presentation will also be followed by Kathleen Lava, PhD, Extractables and Leachables expert at Nelson Labs Europe. Hi there, Kathleen. Thank you for joining us as well.
Hello, everyone.
It's a pleasure to have you. So my name is Marcus, and I will be today's moderator. But before we begin, I'd just like to inform our viewers that there will be a live question and answer session after both presentations before the end of the webinar today. So audience members, please feel free to send in as many questions as you like at any time during the webinar via the questions tab, and that's located directly below your webinar screen, and we'll go through these at the end. Please also state in your questions which presenters they are for. Now, please allow me to introduce our first presenter today. Jason Creasy is a graduate analytical chemist. He has recently set up as an independent consultant providing advice in the area of extractables and leachables after working for GSK in the area of extractables and leachables since the mid-1990s. Over that time, he has seen demand in the area grow exponentially, and Jason has held roles in increasing seniority relating to the support that GSK has given to extractables and leachables. Before setting up Maven ENL Limited, he was director of a team of analytical chemists who are responsible for GSK's global R&D support for ENL activities across a wide range of product types and modalities. This included biopharmaceutical and small molecule, including inhalation, parenteral, and cell and gene therapy use. He has had the pleasure of commenting on PQRI guidelines on extractables and leachables for GSK and extractables and leachables section in EMEA guidelines on inhalation and nasal products, and co-authoring a chapter within a book entitled Leachables and Extractables, Handbook, Safety Evaluation, Qualification and Best Practices Applied to Inhalation Drug Products. Jason has been a member of several external groups concerned with the development of best practice guides for extractables and leachables issues, and these include the IPAC-RS Material Working Group, BPOG, and continues as scientific advisor to the Extractables and Leachables Safety Information Exchange, otherwise known as ELSI. Currently, he is working and commenting on risk-based approaches to ENL requirements, which he hopes will form a part of the ICH guidance in a not too distant future. And with that, I'll now like to hand over to our first presenter today. So welcome, Jason.
Thank you very much, Marcus, and thank you everybody for your time today on this webinar. So today I will be speaking on the subject of future trends to control risks from leachables. The risk from leachables is a subject of interest for around 30 years now. So you might be asking, why am I speaking on it now and talking about future trends? Well, last year work started on the first international guidance document for this topic area, ICH Q3E. So it seemed a good time to reappraise how leachable studies risk is studied and look forward, not back. It's my belief that the risk from leachables needs a specific risk management process, and that's central to my presentation today. It's also the case that the interest in leachable risk control continues to grow, and now with more complex systems and modalities than ever seeking regulatory approval. These approvals need a consideration of leachables, both the effect they have on safety and sometimes on the quality of the drug product. So now I believe is the right time to agree the steps that should be in place and the activities needed to achieve leachable control. So the contents of my presentation today are, as you can see them on the screen. I'd like to discuss the following areas. So what is leachable risk? What do I mean by risk assessment? And what is risk control in the context of this? I also want to differentiate this from the safety risk assessment of leachables, because this is different. And I also want to go through how you might optimize the mitigation of the risk through risk control. So with that, I'll be looking at two key activities: identification and quantification, and linking them to the control of the risks from leachables. And hopefully we can see together how these activities are conducted and how they might be done in the future, and both how these activities influence the outcome with regard to a risk from leachables. So before I get into the substance of my presentation today, I just wanted to spend a little bit of time illustrating that much of my presentation today will be looking at a variety of process steps. And before getting into that detail, I also wanted to remind everybody that every process has its inputs and its outputs, and leachable risk management in that regard is no exception. In fact, there are several process steps within leachable risk management, and each of these has their own inputs and outputs. So understanding that is very important. Also, ambiguity and uncertainty are both major components of the risk from leachables, and this can have a significant effect on the process. And once we remove these, the actual risk is likely low. So it's the quality of the inputs and the clarity of the process which influences the output. And uncertainty can affect inputs, process, and therefore output, as I shall demonstrate. So, coming to knowledge and understanding, these can be built from a variety of sources, and some of these will be obviously more impactful than others. And thus this information must be weighted accordingly in the leachable risk management program. I've listed some of the types of information sources here, which can be collected and will have an impact on the knowledge of the leachable potential. So starting with a general description of the material. This might be of minor use, but it could be suggesting a typical leachable present based on perhaps your prior experience, or indeed the supplier may be able to add a variety of information to the understanding of the material itself. So I would certainly suggest that everybody works hand in hand with their supplier on this front. So what the supplier may also be able to give to the process is some list of compliance statements. They might not relate directly to a pharmaceutical process, but even if it's something like regulation of food, they can be useful in approximating what the risks might be. So the second point here that I'd like to turn to is USP chapters. USP have a variety of chapters that have some application with regard to extractables and leachables. So the first of these is USP physiochemical testing chapters. So that would be 661 and 381. These are actually, at the moment, providing some basic standards and tests that provides some understanding for the risk from materials. But both in the future have planned to have more of a focus on the risk directly from extractables and leachables. So it will be interesting to see how that develops over time. Turning to the biocompatibility chapters, so USP 88 and USP 87 have been around for a very long while, and again are going to be subject to some review in the future. So USP 87 is an in vitro biocompatibility chapter, and USP 88 is the accompanying in vivo test. But they both aim to look towards biocompatibility with regard to the substances that may leach from a material. Further biocompatibility testing is described under ISO 10993. This is, strictly speaking, aligned with medical devices, but there's a lot of overlap between medical use in devices and pharmaceuticals. So it's well worth an understanding of the chapters, even if you're not working in those strictly in the area of medical devices. European pharmacopoeia testing also provides information, It tends to be focused more readily on plastics than elastomers, but nevertheless, any place is a good an additive piece for the risk itself. So finally, there's the obvious option of previously conducted extractable or leachables or testing. All of these listed here are potential inputs into a risk assessment process. So I'm going to move on to that risk assessment process and illustrate what I mean. So I'd like to present my view on what an effective leachable risk management program looks like. Overall, it should have a defined process, and individually there should be effective process steps which all look towards the management of the risk. As I remarked before, risk is scored and perceived potentially much higher than the actual risk, particularly if there is a lack of knowledge and understanding. So the aim of this process is to formalise the collection and evaluation of the information as it relates to leachable risk. And in doing so, you can act on the uncertainty and give an opportunity, therefore, to score the risk more accurately. And each of these individual steps has that at its heart. The graphic here describes a process that is actually very similar to the current ICH process Q9, which is applied to overall quality risk management, with risk assessment, risk control, and risk review. As I've demonstrated here, I believe this can be easily adapted for leachable risk, with one or more risk assessments done as the project moves forward, and the materials, be they used in packaging, combination devices, or manufacturers screening for the leachable risk in a manufacturing system, the same process can be applied. Additionally, once a product is developed and approved, the established risk assessment can act as a baseline throughout the life cycle management, enabling you to aid in the assessment of changes which may affect leachable risk. Thus, this process has the possibility of being repeated several times as the product is developed, approved, and managed through its life cycle, accounting for changes which are introduced and their potential to affect the risk from leachables. So let's look at each step in more detail, beginning with material screening. Material screening is a first step in the risk management process. It's a process that has its starting point when the drug product is being initially developed. The material screening and selection is an opportunity to collect information surrounding the materials that will play an important part when the risk assessment is undertaken. Material screening might itself involve a risk assessment step, but perhaps a less formal one than the one that follows it. But it's a chance to perform an initial assessment of materials to establish the potential they may have as being a source of drug product leachables. This potential is dependent, of course, on both the material and the environment in which it's used, but the selection of good quality materials with the knowledge of the substances that may be present is a first step in allowing the potential rejection of unsuitable materials. An unsuitability may actually mean an inability to gather the necessary information from suppliers. As I stated before, collaboration with suppliers is a very, very important particular point, and it's particularly important when there's a likelihood that those materials might be a source of drug product leachables, as this information will be necessary and useful later when the product is in the clinic or indeed as a marketed product. You can also take the time at this point to create an entry into the drug product QTPP, or the Quality Target Product Profile, and capture the attributes as they relate to the material, defining the requirement for the material quality as it relates to leachables. I believe this is a very useful exercise because it allows all concerned in the project to really focus on where leachables might come within the risk for the project, and then design a program around that to eliminate them or limit them as much as possible. So I'd like to move on now to the next step in the process and look more closely at the risk assessment process. The risk assessment process is the heart of any risk management approach. And as illustrated here, it is subdivided into 3 steps: the risk identification, risk analysis, and risk evaluation. And each of these has its own purpose to play within the risk management process. I come to this point now that risk assessment that people may be more familiar with in terms of extractables and leachables is a risk assessment that up to now has been somewhat appropriated by the focus on safety assessment. But what I hope I can show is that a wider risk management process is also necessary and useful before any testing is planned, and that testing associated with a safety risk assessment for the discovered substances. Thus, the risk identification, assessment and evaluation in this context is referring to the assemblies, components or materials and the environment they sit within as part of the manufacturing process or packaging system or the combination device. That is, that the focus is on the input materials and not on the actual output substances. It's a proactive and not a reactive process. So I hope that differentiates it in people's minds from the safety risk assessment process. But I will be returning to the safety risk assessment process a little later in my presentation, and perhaps that might illustrate it a little bit more. So I'd like to look at each of the processes that I've just described in a little bit more detail now. And the presentation slide that I have up right now has each of these within it. But what I've chosen to do is actually subdivide the risk identification here. And you can see, as suggested by its name, it's an identification and a classification of how and where the risk occurs. It's a necessary first step, and it clarifies the requirement before moving on to risk analysis. And I'm very much of the belief, if the risk is poorly described in this step, it's much harder to do the subsequent parts. So time and care in this step is absolutely rewarded later on. And what you can see illustrated by this slide is I've chosen to list it in the format of a failure mode. And that failure mode has a cause, a risk event, and an effect associated with it. And that can be transferred into a sentence that is useful in construction of the risk the identification of the risk. So because of, there is a risk that resulting in, and the resulting in could relate to safety, and more often that is the focus. But there's nothing to stop it actually relating to other properties that may be equally important to the project, such as product performance or market acceptance in some way, shape or form. And here you will see an example here with the elastomeric stopper. So the elastomeric stopper, the risk event is describing here is the long-term contact with the drug product formulation. But it illustrates here that there are several potential failure modes that could be described for a single material. So whilst I've illustrated it with the long-term contact, there may well be another 2 or more risks that you could describe for that material. So this is why I think it's very important to sit and clearly clarify the requirement, because the risk profile might change depending on the failure mode that you describe. So here it is the risk from the substances from the stopper that will leach from the stopper and into the drug formulation. And the effect on the patient, therefore, is that these will be dosed to that patient, taking the patient in the prescribed way. So once again, here you have the opportunity to be really, really descriptive and predictive of the risk itself. And you can therefore end up with risks that change in magnitude as a result of what you write here. And moving on to where the risk occurs. The risk occurs, obviously, it has the potential to occur in a number of different ways. So taking the time to focus on that, if it's a manufacturing system, perhaps look at a process flow within the manufacturing system, or if it's a container closure system, clearly having access to drawings and understanding the mechanisms whereby the leachables can move from one part of the system into another, all add to the richness of the exercise, and you're more likely to identify the risk correctly and accurately. So moving on to risk analysis. In risk analysis, the risk is scored, and the method of scoring here could be equally qualitative or quantitative, but in either case, it's important to define the terms and the scoring scheme before you start the exercise. You can't make the scores up and change the scores as you go through this. You have to pre-score, set a criteria for the scores before you start the exercise, and then score against them. And the scoring here, I would suggest, is on the basis of risk severity and the probability of that risk occurring. And here too, we can account for any knowledge gap, and we can include that in our scoring of the risks. So this is if you like, is the replacement for the detection term, which is commonly included in other risk assessments that use the format that I'm describing here, which is a failure mode effect analysis. So in this slide, what I've illustrated is some of the parts that fall into the categories that I've described. So under the severity factors, I'm suggesting that this is where you would think about what the substances might be in the material, and also the amounts they may be present, and what influence they may have on safety or the quality of the product, depending on how you're writing your risk. And then you would link that obviously to the property of the material as well. And separate from that, then you would consider the probability factors. So things like contact time, contact area, and temperature, and any potential for reduction, along with the consideration of the compatibility of the material with the system in question. And that last component there around knowledge and understanding, clearly there are links here between what you might know about the material. If you know very little about the material, then inevitably you'll be scoring the risk higher than if you know a very great deal. But if you know a very great deal, you may know that this is actually a very risky material. So it can work in both directions, knowing a large quantity of information about the material. But generally speaking, the more you know, the lower the risk it tends to be. So finally, we move on to the process of risk evaluation. And this step is a comparison of the scoring done in risk analysis against the risk criteria of some kind. Now, the risk criteria, again, can vary, but what I suggest here that the best type of ones are simple ones. Either a simple binary high or low risk, or a do or something or a do nothing risk. You could have more elaborate systems, but I personally favour the simpler binary choices over the more complex ones, as the outcome of the risk assessment is then much clearer. Our analytical efforts and associated experimental design can then be focused and delivered not against some nebulous and unfocused risk, but rather now clearly directed at a thought out and identified risk. And this would have some opportunity then to design those experimental programs directly related to the risk that you have established. And that's really where I want to move on to now is risk control. So risk control is the next step in the risk management program here that I'm describing. And risk control itself is made up of two parts. So if we do arrive at high risk that needs to be understood, how's this best done? So we have parts here both talking about risk assessment and risk reduction. Risk exception can actually mean two things. It can either mean that the risk is low and therefore we do not need to do anything further, or we accept that the risk is high and there is no further possibility of further reduction. And in this case, this then leads to the potential, at least, for the monitoring of the risk. And this could be done perhaps in this context through quality control measures of some kind, or simply documenting the risk in that way, and therefore being aware of it. I sort of illustrated that here with the sort of shark on this slide, because I think it's interesting here that the divers sitting in their cage there are certainly aware that there is a risk, so they've accepted that risk. But what they've done is they built themselves a little cage to sort of mitigate and control the risk in the best way that they believe they can. So they still operate with that risk in place. But however, if risk reduction is indicated, then at this point we are talking about what options there might be. And that's where I want to go with my next slide. So risk reduction options have a variety of choices that can be made. There are options, but two of these actually physically reduce the risk, whereas the third one really comes back to an idea that it doesn't reduce it, but it can better inform the risk. So you get a more accurate picture of what the risk is, so less uncertainty, as it were. So you could choose to eliminate the risk. If you've established that the material or the process is responsible for the risk, you then have the opportunity to replace it or change it. Of course, this would mean that you would have to assess the new material or the new process, but it's a good approach if the risk is very high or you're very early in the project. But it's obvious that later in the project, it's much more complex and much harder to occupy, to change or the material or indeed the process. But I just want to make sure people, don't dismiss the idea of these, because ultimately they are options, and it is something to think about and explore. But likely, what we turn to is this third idea here of gaining more information. But as I said before, what this actually is not really an ability to reduce the risk, but actually perhaps to accept that we have overestimated the risk, normally due to some uncertainty with regard to the risk. So we've reached the option now where we think about how we're going to gain that more knowledge and understanding. And that perhaps then does lead us to extractable or leachable studies. And at this point, they really do start to add value. Doing it at this point, you've now clearly defined the reason for the study, and you can design and target it accordingly. Whereas I think without this, you are potentially launching studies without doing the risk assessment beforehand. You run the risk of those studies being inappropriate and not really giving you much direct reduction of a risk. And actually then demonstrating that in your studies can be harder. So moving on then to these studies, I just want to describe them in more detail. So this slide is entitled Risk Reduction, but really what it illustrates here is a general process for the operation of either an extractable study or a leachable study. And in this part here, I'm not really differentiating between whether it's an extractable study or whether it's a leachable study. I think the unit operations are the same, and in practice you're doing similar activities. So there is a creation of an extract, there is an analysis of an extract, followed by a detection of the substances. We then come to two very important process steps, which are both inputs into the safety risk assessment process. That is the practice and the process of identification and quantification. And these are the two areas that I want to focus on for the remainder of my presentation, as these activities have developed significantly over the time that people have been considering extractables and leachables. And I think it's important, therefore, to spend some time thinking about them and talking about them. So, like an iceberg, identification of extractables and leachables can start from a position of ignorance as regards the true nature of a substance. And even with identification from a mass spectrometer, which is one of the things that is typically done, you may be left with not enough to propose an identification. And what I'd like to do is just illustrate here the terms that are actually now in USP 1664, and hopefully we can they will be refined further as we move forward. These terms are subdivided here, as you can see, into confirmed, confident, tentative, and unknown. And a confirmed ID invariably requires a reference substance, so that when we propose an identification, a chemical structure can be ratified. Without the reference material, you may have confidence from your pooled information to propose a structure, but there's still a chance that you may have an incorrect one, since the spectroscopic information can be misleading, particularly if you rely only on library spectra without some sort of orthogonal data set, such as a max retention time or another technique to provide data. Therefore, the tentative identification, or indeed unknowns, are normally unsuitable for safety assessment, as the assessment is going to be made on the assumption of an accurate structure. And changes in the chemical structure, even with the same mass, can have a profound change in the output of a safety assessment. An illustration of this might be isomers that are metabolized very differently in the body. So if you do have once you have a confirmed or confident ID, you're much better placed to move forward on and think about quantification. So it's obviously a very wide subject, and it covers many topics which I could talk at length about. But today I really wanted to focus on one which I think is a major one in terms of future direction, and one which has certainly been a discussion point as far back as the original 2006 PQRI document. When we use LCMS or GCMS, any quantitation will need a response factor to correct for variation in detection of response. And the magnitude of the response from substance to substance can vary. That variation will not be a significant problem if there is a standard reference substance run alongside the extract. But I'm sure, as this audience understands, it's not uncommon to be unable to run an exact reference substance. Therefore, in most ENL analysis, an internal standard of some kind is run alongside inside the extract, and the relative responses of the substance of interest are produced. But as you can see from the table at the bottom here, which is actually reproduced from an excellent paper from Nelson Labs that I recommend everybody to read, there can be quite a variation in the magnitude of relative response factors, and particularly if you're conducting the analysis via LCMS. So why does this matter? Well, variation introduces error into the measurement compared to the true value, which is particularly important at or near the reporting threshold when this becomes a decision point for safety assessment. So approaches to this have varied. The original 2006 PQRI document actually offered two solutions, but over the years, the more difficult one of the two has been a little bit neglected, but it does have important implications. The original PQRI offered the idea of identifying everything, and obviously that is sometimes possible, but not always. And the second option was to apply an uncertainty factor. Now that uncertainty factor was either to be 50%, the PQRI suggested, or the uncertainty factor was derived from a relative response database, whichever was the greater. And this is what was written in the PQI guidance of 2006. But people being people, the 50% relative uncertainty factor was adopted readily, since it was a much simpler option than the other parts I've just mentioned. And this factor is actually quite, indeed, very, very appropriate for most GCMS analysis, which is what dominated ENL at the time. But as we move forward now, and LCMS has grown in capability and application, it's more and more necessary to pay attention to the second part of the PQRI suggestion, and that is an uncertainty factor based on an RRF database. But this is not a trivial undertaking. An RRF database must be carefully curated and statistical analysis applied to derive the uncertainty factor. A small and unrepresentative data set will not lead to an appropriate correction of the RRF. And I'll remind the audience that a correction to the RRF is only necessary if the peaks are unknown. So an alternative approach, as I've just mentioned, is actually reliant on that more complete understanding of the system. So turning to the safety assessment piece that I said I would return to, when an accurate identification and quantification have been achieved, this can then be passed to a toxicologist for a safety assessment to be performed. However, it's again not a simple process it could be, and is potentially impeded once again by a lack of information. A toxicologist will be looking to establish if the amount of exposure has adequate margin of safety compared to a limit of some kind. And that limit is dependent on what we know about the substance in question. The best case scenario is that the substance in question has had a lot of toxicological studies undertaken, i.e. it's a data-rich substance, and then a clear calculation of permitted daily exposure can be made from this. And the measured exposure can be compared against this, and hopefully you then have your justification for acceptance. But alternatively, there may be little known about the substance and its toxicity. And in this instance, a much more general limit must be applied based on its structural features and predicted toxicology, as well as things like duration of contact. This might also be a read across from some other related substance if perhaps that related substance has more information. And so once again, you see here that the final safety assessment process is made potentially much more complicated and reliant on what we have by way of available information and knowledge. The actual process of doing safety risk assessment has not really had a full agreement on the process, and the allowable thresholds are still being debated. But I hope the upcoming ICH Q3E guidance might provide some assistance here. So I'd like to turn to a summary of the points that I've been through here with my presentation. And what we see here is that the risk control of leachables is highly dependent on available knowledge and understanding. And tools such as mass spectroscopy can be very helpful, but care is needed when assigning both identification and amounts. And it's clear that mass spectroscopy will remain a very important part of activities regarding extractables and leachables. But the future, we really must try to resolve some of these limitations and move in that direction. And hopefully this presentation has discussed some of that. And the linked issue of analytical evaluation thresholds. Once again, future trends must try to resolve the ambiguities that I've illustrated here, with agreement over its application and its practice. But a very clear workflow is yet to be agreed. But once again, I would suggest that ICH could offer a route to this. And finally, I come to risk assessment once again. It will remain a key process step, but we must resolve the inconsistencies in how it is conducted and agree with all stakeholders a common method and universal agreement on how that might be applied. And I hope that all of these points will continue to be developed and improved. And at that point, I'm going to hand back to Marcus for the next presentation.
Great. Thank you very much for that great presentation, Jason. And just a quick reminder before I introduce our next presenter, for our new audience members, there will be a live question and answer session after this next presentation before the end of the webinar. So please feel free to send in as many questions as you like at any time. Just please state in your questions which presenters they are for. And with that, I'd now like to introduce our next presenter. So I'd like us all to welcome Kathleen Lava, who successfully completed her PhD in chemistry from the University of Leuven in Belgium in 2012. Afterwards, she performed postdoctoral research in a team of Professor Richard Hugenboom at Ghent University. In 2016, she started at Nelson Labs in Europe as a study director for Extractables and Leachables. She is part of a team specialized in large volume parenterals, ophthalmics, and disposables, with focus on ophthalmic drug products. And with that, I'd now like to hand over to our next presenter. So welcome, Kathleen.
Thank you, Marcus, for the introduction, and thanks to all of you for joining this webinar. I will use the remaining 20 minutes to tell you more about extractables and leachables in ophthalmic drug products. To get an idea about why we are testing ophthalmic drug products for extractables and leachables, we have to take at hand the existing guidelines. As you may see in this table, both FDA and USP 1664 classify ophthalmic drug products as high risk, given that there is a high degree of concern with respect to the administration route. The table you can see here is a screenshot of the USP 1664 document, which is an updated version from the original FDA guidance. In the original FDA guidance for packaging human drugs and biologics of 1999, extractables testing is considered for ophthalmic drug products, but not imposed. More recent presentations by FDA representatives clearly state the need for leachables monitoring in ophthalmic drug products. And this point of view is also shared by the PQRI, who has classified ophthalmics together with parenterals, and testing should be performed accordingly. However, in terms of packaging and route of administration, it cannot be denied that there are significant differences between parenterals and ophthalmics. The administration is topical versus parenteral, and the dosing volumes for ophthalmics are generally extremely low. Also, the toxicological endpoints that should be considered are different, with sensitization and irritation being more relevant for ophthalmic drug products. This being said, today I would like to focus on the specific nature of the packaging system for ophthalmics and the consequences for extractables and leachables testing. So how does the packaging system look like? In fact, there are two main presentations: multi-dose eyedropper systems and unidose blow-fill seal ampoules. In most cases, a label is directly applied to the primary packaging, and they are distributed in carton boxes or first packed into an over pouch. The base material used for the production of the primary packaging is polyethylene or polypropylene. These materials can contain antioxidants such as Iriafos 168 or Irganox 1010 and some acid scavengers. Additive-free grades are also widely available. Extractables that can be expected from these materials are degradation products of the polymer, such as aliphatic hydrocarbons, ketones, small organic acids, antioxidants and their degradation products, and fatty acids. But all in all, these are materials of high purity that do not lead to a high amount of extractables. And please also note that the majority of ophthalmic drug products are aqueous-based, and chemicals with low water solubility will not easily end up in the final drug solution. Now let's look at an ophthalmic drug product we have recently tested. This was a blow-fill seal ampoule with a label attached to it and packed into an over pouch. Prior to leachables testing, extensive extractables testing was performed. not only for the ampoule, the primary packaging, but also for the label and overpouch, the secondary packaging. The reason why we are performing extractables testing prior to the leachable study is to get an idea of the identity of the compounds that need to be monitored during the leachable study. For the overpouch, the GCMS chromatogram of the ethanol extract in this example showed a number of peaks. Those were identified as cyclic esters. Given that these esters were already identified as potential leachables in the very beginning of the study, we were able to closely follow up them during the leachables testing. And indeed, when we looked at the leachables profile, the only leachable that was detected above 1 ppm was one of the esters originating from the overpouch. This kind of leaching from secondary packaging is well recognized by the US and European authorities. Specifically for semi-permeable containers made of LDPE, they are asking for scientific proof that secondary packaging components are not compromising the product's safety. Please note that only the label and ink are mentioned in the guidelines, but also testing of other secondary packaging components such as pouches should be performed. To avoid surprises during drug product development, we recommend to take into account these materials at an early stage of the packaging's safety assessment. Let's take a closer look at the label and the overpouch. The label is mostly directly attached to the primary packaging, and leachables can originate from the paper, adhesives, varnish, and ink. Typical extractable compounds are curing agents, surfactants, and monomer residues such as small acrylates. Also, nitrosamines may need to be considered when dealing with ink-containing packaging components. The overpouch is not directly attached to the primary packaging, but high temperatures applied during sterilization in the overpouch can cause significant leaching into the solution. Typical extractables are cyclic oligomers and bislactones that are used in the binding layers of these multi-laminated foils. Before going into the implications of leaching from secondary packaging with respect to the ENL study design, I would like to note that also tertiary packaging components such as the unit carton and prescribing information leaflet are getting more and more attention. Similarly as for the label, paper, ink, and varnish can contribute to the leachables profile. From experience, mostly abiotic acid-related compounds are detected in the final drug product at significant concentrations. LCMS would be the most suited technique to pick up these compounds, which is remarkable since testing for tertiary packaging components is often limited to screening for volatile and semi-volatile compounds by Headspace GCMS and GCMS. So how should an extractable study be designed to take into account all the different packaging components? During the PDA workshop held in Washington in 2018, it was announced that the PQI working group will make a clear distinction between ophthalmic drug products and parenteral drug products with respect to extractables testing. As for parenteral applications, material characterization is still recognized as a valid approach for extractables profiling of eyedropper systems. To this end, each component is separately submerged in the extraction solvent and heated in a closed vessel. Using aggressive solvents and harsh conditions, you will obtain the most complete list of chemicals that can be withdrawn from the material. Typical solvents used for ophthalmic applications include water with appropriate pH, and isopropanol or a mixture of isopropanol with water to cover for polarity modifiers such as surfactants. Nevertheless, testing each component separately under harsh conditions also poses some challenges. For example, if you extract a label or overpouch by submerging it into isopropanol or ethanol at elevated temperatures, you will end up with a very extensive list of extractables, as can be seen from the chromatogram on the left side. Since it is too costly and time inefficient to monitor all these compounds, it can become rather complex to make a relevant selection of target compounds towards the leachable study. Another challenge is dealing with complex multi-dose eyedropper systems that are often used for preservative-free ophthalmic solutions. These droppers consist of a number of smaller components, for example, filters, valves, springs, gaskets, and testing each of them separately using extreme conditions is less representative to the final use. In those cases, PQRI considers a simulation study to be more appropriate. Here, the packaging system is extracted by filling the labeled container with a suitable extraction solvent, followed by incubation at elevated temperatures. Ideally, the unit carton and information leaflet are also included into the setup. By removing the direct contact between the extraction solvent and the secondary packaging, a more realistic extraction profile is obtained, and all components are considered in one single extraction experiment. In this approach, you will end up with a list of chemicals that are truly potential leachables. Now, to avoid highly volatile compounds from escaping, so to say, during the incubation, it is strongly recommended to use an impermeable incubation vessel during the extraction of the packaging system. The factors that are determining the quality of the experiment are the solvent choice, the temperature, and the incubation period. Solvents in the simulation study are more product-like, preferably a placebo, but buffer solutions and isopropanol water mixtures are also being used. In other words, the solvent choice is based on the composition of one specific drug solution, meaning that the generated data may not be very useful for other solutions stored in the same packaging system. To avoid the formation of low-melting LDPE materials, the temperatures should not be higher than 60 degrees Celsius. The duration of the incubation is generally longer compared to material characterization study and can be calculated based on the actual storage conditions using the ASTM standard or, alternatively, the factor 10 rule. Either way of testing, the extracts generated are subjected to a variety of techniques so that compounds of different physicochemical properties can be identified. The most commonly applied techniques include headspace GCMS for the detection of volatile organic compounds, GCMS for the detection of semi-volatile organic compounds, LCMS for the detection of non-volatile organic compounds, ICP-OUS for the detection of elements, and iochromatography to identify anions. Once a full extractables profile is available, a leachable study should be performed to evaluate the impact of the packaging material on the drug product's safety and quality. Whereas extractables testing is performed in pure solvents to avoid analytical interference, a leachable study is performed on the drug product itself. The drug solution can give rise to significant interference caused by the active ingredient or excipients present in the formulation. Now you may wonder, are we not making things more complicated by making this assessment in complex matrices? Well, my answer would be yes, but in skipping the leachable study, one may miss out on so-called secondary leachables, which are leachables that interact with one of the constituents of the formulation. Also, the impact of secondary and tertiary packaging components can be evaluated under real-use storage conditions. Furthermore, authorities are requesting leachable studies as they serve to be confirmative and focus should lie in quantification of the detected compounds. Samples for leachables testing are prepared as per the final intended use, meaning that the drug product should be subjected to the same sterilization steps as the marketed product and all components of the packaging system are to be included. The leachable study is performed over the entire shelf life under real use conditions and/or accelerated conditions. Samples should be taken at different time points, as it is not always the case that the worst-case leachables profile is obtained at the end of the shelf life. Just think of volatile compounds that may have been evaporated after 2 or 3 years of storage. Also, batch-to-batch variability has been observed, which is why some authorities are requested to perform the leachable study on 3 different batches of the packaging system. In order to perform a leachable study, not only a solution stored in the packaging system is required, but also a suitable reference solution should be provided. Such a reference solution allows to differentiate leachable compounds from matrix constituents. A good blank solution is stored in an inert container, for example, a glass bottle with Teflon-coated cap for the analysis of organic compounds, or a grinder tube for the analysis of elements, since elements may leach from the glass bottle. The blank sample must be aged under the same conditions as the contact samples, given that the degradation of the matrix components may otherwise be wrongly identified as leachable compounds. For leachable studies on the final storage container, the blank solution can be sampled at the end of the filling line. Generally, a leachable study consists of 2 parts. On the one hand, the contact solution needs to be screened for unexpected leachables, and on the other hand, compounds of concern as identified in the extractable study should be monitored in a targeted way, or at a minimum, a proof needs to be provided that the used methods are capable of detecting these target compounds in the matrix solution. Evidently, fully quantitative results for leachables can only be obtained using validated methods. For both screening and targeted analysis, it is highly important to think about the level that these leachables need to be evaluated against. As you may see in the chromatogram, a high number of peaks were detected during testing. The green line, however, represents the level at which compounds are of toxicological concern to the patient. As such, leachable compounds that are detected below this analytical evaluation level do not need further consideration. Although this concept seems quite straightforward, it is not so evident for ophthalmic applications due to the fact that there is no consensus on the levels to be applied. The main reason for this is that there is insufficient toxicological data available with respect to sensitization and ocular irritation to set safety thresholds for ophthalmic solutions. These toxicological endpoints are different from, for example, the toxicological endpoints that are relevant for parenteral applications. Whereas for parenteral applications, a toxicological limit of 1.5 micrograms per day is recommended, FDA presentations have mentioned concentration-based thresholds being 1 ppm as a reporting threshold, 10 ppm as an identification threshold, and 20 ppm as a qualification threshold for ophthalmic applications. In practice, a two approach is often applied, in which the reporting limit is compared to the SCT base limit of 1.5 micrograms per day, and whichever is the lowest is used as a reporting limit for leachable compounds. Before going to the conclusions of this presentation, I would like to shortly discuss some practical considerations related to the leachable study design. Often, ophthalmics are marketed in different presentations with respect to the container size and filling volume. In this case, the leachable study may need to be performed only one, being on the container with the highest surface area to volume ratio. This is mostly the smallest container. Secondly, more and more formulations are containing emulsifiers and surfactants, or a combination thereof, for which more complex sample preparation procedures are required. As a result, more extensive method development may need to be performed prior to the start of the leachable study. Lastly, the timing of testing is often an issue, and one should take into account that extractables and leachables testing consists of a number of consecutive steps, which also brings me to my final slide. In order to set up a well-designed extractables and leachables study, the following steps need to be taken into account. First, the components of the packaging system that may contribute to leaching need to be defined. Subsequently, the extractables profile of these components needs to be determined, and based on the extractables data, components that are of toxicological concern have to be selected so that they can be monitored during the leachable study. In order to monitor the target compounds, specific analytical methods may need to be developed and validated. Once the methods are in place, a formal leachable study can be carried out on the contact samples that are stored under simulated use conditions in parallel with screening for unexpected leachables. And finally, the leachables that were detected in concentrations that are of toxicological concern will need a toxicological assessment. In total, the full battery of testing has a lead time up to 34 months, meaning that it is a good thing to plan ahead. And with this, I hope you have enjoyed this presentation. My final take-home message would be that secondary and tertiary packaging components are to be taken into account as possible sources for leachables, and that the qualification of the packaging system should be initiated early in the development process. If you need any help with your ENL testing, do not hesitate to reach out to us on the email address shown in the slide. Also, it is certainly worth having a look at our website, as we are offering a variety of microbiological tests, as well as the website of our sister company, Sterilogenics, should you be interested in sterilization solutions. Thank you again, and if there are any questions, feel free to ask them.
Great. Thank you very much for that presentation, Kathleen. Just going to bring us back to the presenter screen. There we are. So welcome back. And yes, thank you very much. And just to resonate what Kathleen said there, please don't hesitate to send in any questions. And if you want to follow up, then please feel free to send an email to the one that Kathleen has made available. But we're about to jump straight into our Q&A session now. You can still send in your questions if you want to. Please just continue to mention in your questions which presenters they are for. And our first question to kickstart things off today will go to Jason. So Jason, can you please explain why should we be placing importance on risk identification by documentation from suppliers when this can be mitigated by performing an extractable and leachable study with a device?
I think the point I'm trying to make is that there is no such thing as poor information. All of this information is useful. In defining the risk properly, you have the opportunity to potentially avoid doing costly experimentation, first of all, if you truly believe that and can demonstrate that the risk is low. If you do move on and decide that you need to do an extractable study or a leachable study, the risk assessment process helps you with the study design. So it's not a case of either or. It's a case of these are complementary things. And what I would also say is that those studies can feed into the next risk assessment. So when you do a product that is very similar to the one that you have previously done, that's part of the risk assessment process. You have the extractable and leachable study, so you would consider the risk to be low if you have the appropriate information already generated. So they're complementary. It's not really an either or a replacement for.
Great, thanks a lot for that, Jason. Any additional comments from Kathleen before we move on?
No.
Okay then. Great. Our next question then will go to Kathleen. So, which of the extraction approaches are best received by the authorities? A simulation study or material characterization?
Yes, thank you for that question. Both filling and incubation and submersion are equally valid approaches. The key things to take into account for performing extractable studies are the extraction solvents. Are they relevant to the final drug product? Let's say if you have a drug product with pH 4, then only performing the extractable study at pH 7 is not sufficient. The extraction conditions are also very important. They should cover the real use conditions. And then the evaluation level is also something they look at for both of the approaches.
Great, thanks a lot for that. Any additional comments from Jason before we move on?
No.
Yeah, okay. Our next question is for Jason. What do you think is the biggest issue which ICH-Q3E can help resolve? Help solve.
I think it's harmonisation and clarification. I mean, maybe I'm getting two bites of the cherry there, but really, what I think is one of the biggest issues at the moment is that people have conflicting ideas and advice and responses coming from various places. And what we hope from, well, I personally hope from ICH is that there will be a consensus and a harmonisation coming out of that, so that people can have confidence in what they need to do. It's not the case that people are trying to, avoid doing things. They simply simply want to do things and know that they are the right thing, so they don't have to do something different. At the moment, it's a bit of a there's a chance that they do what they think is the right thing, and they still are told, no, it's not the right thing. So that's what I hope from ICH, that there will be a clear consensus and harmonised approach that everybody can get behind and do the right thing with.
Mm-hmm. Great, thanks a lot for that. Any additional comments from Kathleen before our next question?
No.
Okay. Our next question for Kathleen then is, are there any recommendations on how to select target compounds based on extractables data that are to be monitored in the leachable study? What if one of the detected compounds could not identify the extractable study?
Yes, so if you have to select target compounds for the leachable study, obviously the compounds that are present in concentrations that are of toxicological concern, that means that the concentrations that are above the AET, they should be selected to perform the leachable study. If you have unidentified compounds in the during the extractable study in high concentrations, my recommendation would be to identify them so that we can monitor them during the leachable study.
Great, thanks a lot for that. Any additional comments, Jason, before we move on?
I think I just reinforce what was said there about identification of unknowns. I think if you're going into a leachable study with large quantities of unknowns, it's very, very difficult. So I think you've really got to try and identify the major compounds wherever possible.
Great, thanks a lot for that. Our next question for Jason then is, what sort of leachable items would you include in the drug product QTPP that you mentioned in your presentation?
Yeah, that's a good question, that one. It obviously is very dependent on the dose form that you're designing for. Each dose form will have some unique characteristics, but I think generally what you're trying to do is avoid those high-risk scenarios. So you're trying to avoid situations where you have materials that contain substances that are of concern, either from a safety point of view or a quality point of view. And I guess that's the way you would almost have the kind of two different types here. You would have what is it about my product that might affect the quality of my product? So, for example, if you had a product that you knew was very sensitive to metals, for example, then you would want to be extra certain that the metal content was appropriate for them in the materials that you have. But then you have the more generalized thing, I suppose, around safety. So you would want to avoid products that are known sources of unsafe compounds. So an obvious one that I think Catherine mentioned in one point there was nitrosamines. So avoiding a system that has the elements that can generate nitrosamines seems like a very good place to be, rather than having to retrospectively go back and test that they don't have nitrosamines present. So trying to eliminate the possibility of these things by quality by design, as it were, is the whole emphasis around this, what you would put into that QTTP. So avoid very complex formulations where you're likely to generate lots and lots of leachables. Avoid incompatibility in terms of the materials that you would choose. So choose materials that you know are likely to survive in the formulations that you're creating for. Those are the general principles. The rest comes very, very project specific, I think.
Great, thanks a lot for that. Any additional comments from Kathleen before we move on?
No.
Okay then. Our next question for Kathleen is, the turnaround time for obtaining results is relatively long. Is there any way to reduce the timings?
Well, ideally, every step is performed after another step has been finished. For example, if you want to go to the leachable study, first you need the results of the extractable study. You have to make an evaluation of the extractables data, and then only you can what you have to look for in the leachable study. So in that sense, it is not a really good idea to try to in some way perform these steps in parallel. But often what you can do is generate data at 6 months, accelerated aging conditions, and the authorities accept these data for submission. So this is something you best verify with someone from the regulatory bodies before submitting.
Great, thanks a lot for that. Any additional comments from Jason before we move on?
I guess I just come back to that knowledge management piece for me. You can potentially accelerate those studies if you've studied them before, or your supplier tells you all you need to know. So you potentially can do a little bit more in parallel if you've got that prior knowledge, I suppose. But if you know nothing, then.
Yes, indeed. And sometimes the supplier also has extractables data available. So that's also always worth checking before you start extractables and leachables testing.
Great, thanks a lot for that. Our next question then goes to Jason. What is the difference between risk assessment and safety assessment, and also margin of exposure and margin of safety?
Well, as I indicated in my presentation, the risk assessment process here for me is a risk management process that is lots and lots of individual process steps. So lots of inputs, lots of outputs. Whereas safety assessment is focused is one of the processes within the risk assessment process that I've described. It's one of the tasks. The general focus of the risk assessment process that I'm describing is centered around the materials, really. So the componentry or the various parts that the substances would come from, whereas the focus of risk assessment on a toxicological point of view is from the substances themselves. There is that difference there between a focus on the materials and how they might have a risk from leachables, how leachables may be present or not, and once you've done your leaching, you have substances that are then assessed by the safety assessment. So the next part of that question was around what's the difference between the, sorry, what was that? The difference between
safety assessment and also margin of exposure and margin of safety.
Margin of safety. So the margin of safety is simply a term that is used to suggest what is the allowable limit in a unit, let's say micrograms per day, and what is the measured unit of exposure, how much you found. So if you found less than the allowable limit, then you have a positive margin of safety. But if you go beyond the allowable limit, you have a negative margin of safety. So the aim would be to have, obviously a positive margin of safety to remain within the allowable limit. Now, the allowable limit is predicted from toxicological studies, and I could spend a very long time going through how to calculate an allowable limit, and that's quite a complicated situation. But it's the limit that a toxicologist says is acceptable, basically, and you're comparing your exposure to that, and the difference between them is your margin of safety.
Great, thanks a lot for that, Jason. Any additional comments from Kathleen before we move on?
No.
Okay. So our next question, I think is for Kathleen this time, but please feel free to nominate each other if you think it's more relevant. Can a substance which is resulting corrosion in eye cause systematic toxicity?
I think this is something you would have to check with the toxicologist. I am not experienced in that. I'm sorry.
No problem. Any additional comments from Jason before we move on?
I agree. You need to befriend a toxicologist to get the answer to that one.
Mm-hmm.
Great. Okay. So moving on, our next question, once again, I think it's for Kathleen. So is the correct flow, excuse me, is the correct flow first we perform an exploratory analysis before choosing the internal standards for AET determination? Because how can we proceed in the case of finding several classes of compounds with different ionization factors?
Yeah, so the extractable studies we perform are screening methods, and they are designed to pick up a wide variety of compounds. Once you go to a leachable study, of course, if you go to validated methods, indeed, there is method development where suitable internal standards are selected. If you go for a screening leachable study, then you have to perform some kind of verification to show that the general methods you are using are adequate to pick up the compounds of concern at the toxicological level.
Great, thanks a lot. Any additional comments from Jason before we move on?
No.
Okay. So our next question then is for Kathleen once again. What is your experience with regulatory bodies accepting a well-designed simulation study with analytical clarity in place of an actual leachable study where analytical interference may lead to false negative results?
Yeah, so if you perform only a simulation study, you do not have the effect of the constituents that can react with potentially with the leachable. So this is something you will miss out in not performing the leachable study and only submitting with simulation studies. So I think the authorities are really requesting leachable study unless there is no possibility of screening in the drug product itself. But then you need to prove that.
Mm-hmm. Thanks a lot. Any additional comments from Jason?
I think I would just support what was said there, is that there would be very little point in supplying a leachable study if it gave you incorrect results. You're looking to supply whatever study provides you with the best outcome. So that might be a leachable study or it might be a simulation study, but that's the test here, is which will give you the best outcome.
Great, thanks a lot for that. Our next question for Kathleen, once again, is ODP reporting thresholds, 1 ppm, any official reporting limit?
No, there is no official reporting limit for ophthalmics, and this is something we are looking forward to in the new PQRI recommendations, which are expected, I think, later this year. as I also stated in the presentation, there is just a lack of toxicological knowledge on which compounds are sensitive to irritating to the eye to provide such a limit. So this is something we are really looking forward to, because now it's sometimes a little bit shot in the dark.
Great, thanks a lot. Any additional comments from Jason?
Nothing to add.
Okay then. Now my next question to Kathleen once again. Why performing an extractable study and not directly anticipating a leachable study with clinical simulating condition?
I think we have already had that question before.
Okay, that's fine. In which case, we'll just move on. If you do have a further question on that topic, though, please feel free to send me an email, which I'll make available to you audience members later on. But my next question, then, I think this time is for Jason. I think it's off the back of one of your earlier questions. What if you cannot identify a given unknown? What are the alternatives?
Yeah, I mean, it's a constant challenge to identify unknowns, and there are alternatives. One of them is to try and get some more information around the source. Once you understand the source, you have a possibility of understanding what the unknown might be, or perhaps you can understand what class of compound it might be. You know, there is no simple set of instructions here. It is very much dependent on the situation as to what you may or may not be able to achieve. But not everything is solved through an analysis. The key to identifications of unknowns is the context, really. If you can understand what the source is, then you have the possibility of extracting and isolating the unknown. And if you can isolate the unknown in sufficient quantity, you have additional analytical options. You could start to explore things like NMR, perhaps, that would give you more insight into it. Or you could try some, synthesis routes once you have some direction for those synthesis routes. So there are many things you can try, but they all depend on knowledge and understanding. And ultimately, it's possible that you won't be able to identify it fully, but you might be able to narrow it down to a number of possibilities. And once you have a limited number of possibilities, you could risk assess them all in terms of a safety risk assessment, and say, well, the worst of these would be this, and hopefully that might be acceptable still with a margin of safety. So there are always options. Some of them are more time-consuming and expensive than others, but there are always options.
Great, thanks a lot for that, Jason. Any additional comments from Kathleen?
Yes, indeed, it's like Jason says, it's not because you cannot identify it at first sight that it's not identifiable. You can isolate it, you can use some high-end techniques. We have a team which is specialized in identifying unknowns. So there are definitely other options to identify those compounds.
Great, thanks a lot for that. Our next question is for Kathleen. Can you clarify the information about 1 ppm, 10 ppm, and 20 ppm for ophthalmic products?
Yes, so in FDA presentations, these concentration-based limits are recommended. For the identification, a 10 ppm limit is recommended. For the qualification, that means when you should perform a toxicological assessment, a 20 ppm limit is recommended, and then 1 ppm is for reporting the compound. However, in practice, we see that most of our sponsors are identifying and qualifying leachables that are detected above 1 ppm.
Great, thanks a lot for that. Any additional comments from Jason before we move on?
No.
Great, okay, thanks a lot. Next question, I'll ask to Jason. It doesn't say who it's to, but feel free to nominate each other. What are the recommendation leachable time points for dry powder injections?
They would not be particularly different for dry powder injections compared to any other system. You would want to study at best the period that the shelf life is of the product. You might want to also just study any sort of relevant in-use period as well. So it's dictated by, as with any leachable study, the product characteristics themselves. You could, as Kathleen has already suggested, accelerate that and with regard to temperature, but dry powder systems are not really any different to any other systems, in my view anyway. Others may think differently.
Thanks a lot. Any additional comments from Kathleen?
Yes, I agree. I think you should be testing at the initial time point and then mostly at the 6 months time point, and then every year until the end of shelf life. That's mostly the baseline of testing we recommend to our customers.
Great, thanks a lot for that. Our next question here is for Kathleen. In a PQRI document, there are some doubts that the SCT concept could be applied to ophthalmics. Thus, the hypothesis that threshold principles could be extrapolated from OINDP to ophthalmic solutions, and suspensions lack sufficient scientific support to develop a recommendation. What thresholds do you recommend?
Yes, so for now I cannot recommend any thresholds to use. What we do is we indeed take the parenteral SCT from 1.5 micrograms per day, even though it cannot be extrapolated to ophthalmics. And apart from that, we also use these concentration-based limits, and we take whichever is the lowest. So we are weighting the recommendations of PQRI to which limits to use for extractables and leachables testing.
Great, thanks a lot for that. Any additional comments from Jason?
I suppose the only thing I would add is I would hope that the ICH document would think about this and comment on it. It does seem a really difficult piece because of the lack of toxicological information to support a particular threshold.
Great, thanks a lot for that. Our next question for Kathleen. What would be UF in AET analysis?
Yes, this is something we have implemented, I think, in the last few weeks. The uncertainty factor is determined based on our internal libraries, which contain, I think, 3,000 compounds for GCMS and 2,000 compounds for LCMS. And we have determined it as stated in the PQRI document to be 2 for GCMS and 5 for LCMS. So when you determine the AET, the reporting limit is set at a level taking into account these uncertainty factors.
Great, thanks a lot for that. Any additional comments from Jason?
No.
Okay, great, thanks a lot for that. Our next question for Kathleen. What do you mean method validation for leachable study?
So in a method validation, the precision, the specificity are determined for specific target compounds, which means that you also have you can obtain the exact concentrations of the leachables by making use of standards that are spiked at different concentrations.
Mm-hmm. Great, thanks a lot. Any additional comments from Jason?
I think the method, the validation that you do for leachables obviously has to be fit for the system that you're developing for. So it's very much a case of thinking carefully about the system and making sure those validations are appropriate for that system.
Great, thanks a lot for that. Our next question, I'll ask to Kathleen first, but feel free to pass on to Jason if you feel you need to. What are the pros, cons of doing accelerated temp storage leachable study over real time, and which is preferred by regulators?
So the leachable study has to be performed throughout the shelf life. The advantage of doing an accelerated aging study is that you have data that are representative earlier in the process. But it can happen, for example, if you use accelerated aging conditions, that volatiles may already have escaped, which is not the case if you analyze at different time points over the course of the product storage.
Great, thanks a lot for that. Any additional comments from Jason?
I don't think there is a strong preference from regulators. I would have thought that regulators, though, would want to see real-time data of some sort, but I think they are hopefully realistic enough to cope with both.
Great, thanks a lot for that. Our next question, I'll ask to Kathleen first. Is ISO 1099323 applicable?
So that is the guideline which is applicable for medical devices. But Jason, maybe you can correct me if I'm mistaken.
It's specifically the irritation part for medical devices, isn't it?
Mm-hmm. Yeah. Mm-hmm. So it's not applicable for ophthalmic applications because they are considered to be drug products.
Mm-hmm. Okay, great. Any additional comments from Jason?
No.
Okay, great. Thanks a lot for that. Our next question, I'll ask to Jason, but feel free to pass on to Kathleen. How are you determining uncertainty factor in AET calculation?
This is quite a long answer potentially, but I'm only going to keep it short, and I'm sure Catherine has got an opinion on this as well. But there are a number of ways currently that uncertainty factors are calculated, but principally the concept here is around, first of all, you really are worrying only about when you have things without a reference standard. Once you've got a reference standard, then it doesn't really apply because you can potentially determine directly from a standard what the response is. But where you are applying an uncertainty factor, it should be one that is applicable to what you believe the system, the substance is likely to be. If it's a complete unknown, what you would be wanting to do is to set a uncertainty factor broad enough to cope with the variation that you might see for any given system. So having a very wide database to base an uncertainty factor on is a good idea. There are some sort of suggestions in the original PQRI around what might be applicable for GCMS in particular, the 50% of the AET being a suggested factor. But there's a lot of current debate as to what might happen to those factors over time, and I think there's probably going to be a lot of development of thinking around what an appropriate uncertainty factor might be. But it's basically based on as much information and work that you can do around your system.
Great, thanks a lot for that. Any additional comments from Kathleen?
Yes, we are receiving the same messages that the uncertainty factor of 50% may not be longer considered to be an appropriate uncertainty factor for the methods we are using.
Great, thanks a lot for that. Our next question then for Jason, I think, will be, how do you perform a leachable study for the product in 100% oily product? Oil might mask the leachables in GC-LC chromatogram, and So I just lost it. And is not injectable onto column.
Yeah, I mean, I think this is the case for what was being discussed around simulation studies. So I think Catherine will probably have more to say on this than me, but I think simulation studies in this case would be warranted, or some special attention on sample preparation. But I think simulation studies are probably the better option of the two. But I'll pass that one over to Catherine.
Yes, indeed. So a simulation study can offer a solution, but also the combination of a targeted study in which the sample preparation is optimized to look for specific compounds, which we know they are of concern from the extractable study. And this combined with a simulation study gives you a quite good idea of what is happening in the drug product with respect to leachables.
Great, thanks a lot for that. And our next question, I'll ask to Kathleen this time. So for the large volume parenteral product, example 60 litre per day, the AET could be as low as 0.001 ppd. How do you cope with the method?
Yes, also in these cases, you can do a simulation with a lower filling volume of the bag, which also allows you to look at a lower level. Simulation studies are also an option here. The sample preparation validated methods is a combination of different things. You can try to get a complete idea of the leachables that are will end up in the final drug product. So indeed, for large volume parenterals, it's analytically very challenging to reach the reporting limits or the AET. That is correct.
Great, thanks a lot. Any additional comments from Jason?
No, I just support what's already been said.
Mm-hmm. Great. Okay, thanks a lot for that. Kathleen's next question: with GC-MS2 and LC-MS5, how does Nelson validate the concentration procedure?
So I'm not sure if I'm understanding the question correctly, but we have internal standards that run in parallel with our sample preparation steps to see if the sample preparation we use is adequate to pick up certain target compounds. The uncertainty factors, they were determined based on our library. I have to look into the details because it was just recently released, but the uncertainty factors have been calculated based on the mean deviation of the different response factors we have in our libraries.
Great, thanks a lot for that. Any additional comments from Jason?
No.
Okay, then our next question goes to Jason. Do you think that authorities will accept assessment of drug products just based on calculations of an overall HDE PDE by means of extractable data supplied by the material of suppliers? Or do you think there will always be a request for a leachable study?
I think the issue there is about identifying all sources of leachables. If you are basing it on simply extractable studies, you have to be really certain that you have identified all of the potential sources. So, for example, have you thought about the manufacturing of that drug product? Have you thought about all of the different things that packaging parts that might be relevant, tertiary, secondary, etc. So if you've done that complete piece, perhaps there is the possibility that that might be acceptable in the way that you've described. But the leachables actually make a bit more sense because they're quicker, simpler, and easier to do to get to that point. So I'll come back to my earlier point. I think you'd have to look at what's simplest and best, and sometimes that is a leachable study rather than extractable studies that perhaps might provide an incomplete picture. But I wouldn't rule it out completely. But I think it's sometimes just easier to do the leachable study than anything else.
Great, thanks a lot for that. Any additional comments from Kathleen?
No.
Okay, then. So for our final question today, then, I'll ask to Jason first, but feel free to pass on if you would like. What do you do if the AET calculated is too low to achieve, like ends up being below LOD?
Well, I guess it depends, because the LOD may or may not have taken into account the fact that you might be able to enrich your sample through sample preparation. So if you're, it's a bit like the answer to the 60-litre large volume parenteral. There are a lot of potential things that you could consider. You will obviously have a fixed amount of analytical response for a given amount that you inject on the system. You know, there are some sort of hard limits there sometimes, but there are things like, have you considered large volume injection on the GC rather than a small volume injection that is conventional, because that may then increase your signal to noise and sensitivity. So you've kind of got to get sort of step through this in terms of, have I maximized out what I can do in terms of putting substance onto the analytical method? And there are several ways of doing that. As I said, you can concentrate your sample, you can go to a more sensitive form of detection in selected ion monitoring rather than scanning perhaps, or a different type of detector that may be more sensitive. So there are with all things analytical, there are always alternatives. Whether they are sensible in terms of time and cost, you have to judge, but there are always alternatives.
Great. And any additional comments from Kathleen?
I indeed think that you have to make a distinction between the screening for unexpected leachables, for which it may be difficult to reach this AET if it's very low. Then you should go to a simulation study. For targeted leachables, the methods are specifically developed to look at the AET. So I have rarely encountered studies where we cannot monitor targeted compounds at a required level. And that is indeed, as Jason said, you have to optimize the methods, the preparation procedures in order to be able to look at that level.
Great, thanks a lot. So lots of questions today. Thank you very much for sending them in. If we didn't get around to answering all of your questions, then don't worry. You can still send them over to my email address, which is currently displayed below your webinar screen at the moment. My email is marcus.osei@biopharma-asia.com. And if you send your questions over to me, I'll try to forward them on to our presenters and hopefully get those answers back to you. But before I finish today's webinar, I'd just like to ask our presenters if they have any closing remarks. We can go with Jason first.
No, I'd just like to thank the organisers, and it's always great to hear high quality presentations. So I'd like to thank Nelson Labs for their contribution to this too. And I always thoroughly enjoy hearing all the questions from people. So keep sending them in.
And over to you, Kathleen.
Yes, Jason, also, thank you for the interesting presentation. Thank you to the organizers for giving me the opportunity to present today. And thanks to all of you for joining the webinar. Great.
And I'd like to also take this opportunity to thank our presenters. So once again, that was Jason Creasy, Managing Director at Maven Extractables and Leachables Limited, and Kathleen Lava, PhD, Extractables and Leachables Expert at Nelson Labs in Europe. Thank you, both of you, for sharing your knowledge and your expertise with us. And lastly, I'd just like to remind our audience members that you can view this webinar and other webinars on demand by visiting our website, which is biopharma-asia.com/on-demand-webinars. And if you're watching this webinar on demand, like I said before, please feel free to send your questions over to me at my email address, marcus.osei at biopharma-asia.com, and we'll try to get those answers back to you. But thank you, everyone. Thank you, Jason. Thank you, Kathleen. And I hope you all have a great day. Take care now.
Thank you.
Bye-bye.
Bye.